Hi everyone, welcome to your lecture on the ear. So, this is the cranial nerve that will innervate inner ear structures. Cranial nerve 8, vestibular cochlear nerve. That's the innervation there. And there's two parts. There's a vestibular branch and a cochlear branch that detect different sensations. Vestibular branch, cochlear branch, as you can see from the picture. And we're going to learn these different structures of the inner ear. So the inner ear was obviously where it is, deep inside the ear, deep inside the petrous region of the temporal bone. That's kind of what we call it here. So the inner ear structure is located in the petrous region of the temporal bone. So that's what we see here. So as we go from inner to outer, we learn the other components of the middle ear and the outer ear. So I'm working our way from in to out. In this little chamber or compartment in here, known as the middle ear, we have this space called the tympanic cavity of the middle ear. Hold on, I always forget something to erase one. All right, the middle ear is a cavity called tympanic cavity. It contains the auditory ossicles. Audio, audition, that's for hearing. Ossicles, ossification, that means these are bony structures. They're super tiny. And they are the malleus, the incus, and the stapes. That's what it says right there. There are three auditory ossicles. Malleus, the hammer, incus, the anvil, and stapes, little horseshoe-shaped bone with the foot plate. And it's stapes that is in communication with the inner ear structure, because at the foot plate of stapes is this one window that it pushes against. It's called the oval window. So stapes is shaped like this, and it has a foot plate. So the stapes foot plate and pushes against the the oval window of the inner ear. Foot plate in contact with the inner ear structure via the oval window. Now it's called the tympanic cavity because there's a tympanic membrane. I'm going to include that as part of the outer ear. So let's talk about that. More on auditory ossicles later. The outer ear, what we call our ears, in anatomy we say the pinna. It's cartilaginous. It helps to funnel the sound waves to the auditory canal, sometimes called the external acoustic meatus. Funnel sound waves to auditory canal. I don't want you to confuse the auditory canal to this auditory tube, which I also want you to know. I should have mentioned the auditory tube as part of the middle ear because it is in communication with the tympanic cavity. So let me put that as a side note here. Know the auditory tube is in communication with the tympanic cavity. In communication with tympanic cavity. It actually, you can't see it here, it leads to the pharynx or the throat. So it's in communication with the tympanic cavity as well as pharynx. Which is your throat. So it's a very important tube. It's in contact with the ear because of the tympanic cavity association and your throat because of the pharynx. That's why you need ear, nose, throat specialists because the pharynx is in direct communication with your nasal cavities. And so it's important to know where you have communications with each other. That's how infection can spread. So that's why you need ENT physicians. Well, anyways, I should also mention that getting back to the outer ear, I'm going to include the tympanic membrane as a part of the outer ear. So as the sound waves are funneled by the pinna, and you can help, right? It's like when you cup your hand and listen, you're helping to funnel the sound waves to this canal. And all those sound wave vibrations will, the energy will vibrate that tympanic membrane at a certain frequency or hertz, okay? And those sound wave energies are then transferred to this ossicular chain, these auditory ossicles, which will vibrate at the same frequency that you're hearing, and that sound will then be transferred to the inner ear structure, which has the organ of hearing. So, tympanic membrane vibrates at whatever frequencies it's receiving. So whatever sounds you're hearing are vibrating that and transferring it through the middle ear to the inner ear. Okay, so that's the basic idea, outer, middle, inner ear. A little bit more about those middle ear tympanic cavity or auditory ossicles, which are a chain of bones that transfer sound wave energy from tympanic membrane from there to there. And in doing so, let me read what I have on my slide. The tympanic cavity with malleus, incus, and stapes form an ossicular chain, boom, 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 of bones. They amplify and transmit sound wave energy to the oval window. That's worth writing down. Auditory ossicles are in what they call a ossicular chain. That amplify and transmit sound wave energy. To the oval window. There's a round window, but the oval window is the one we're talking about now. So the reason why there's an amplification is, look at the relative size of the tympanic membrane to that little oval window under the footplate of stapes. So you're taking this large area and vibrating, and these bones are all transferring it to a much smaller area. So you're going to amplify it by doing that by about a factor of 20. Some books say a factor of 22 or something like that, about a factor of 20. It's a blank figure for you can study. There's also nerves running in there. That's the facial nerve. You don't got to know that. Just know the tympanic membrane and these auditory ossicles, malleus, incus, stapes, and know that underneath that footplate of stapes is the oval window. Here's another picture of this ossicular chain. As the tympanic membrane vibrates, a slight pressure wave is amplified 20 times as it passes from bone to bone to bone in an ossicular chain. If this ossicular chain fails, the patient will experience what's called conductive hearing loss by about a magnitude of 20 decibels. So just to give you an idea of what 20 decibel means, decibel levels in terms of a number, I give you some examples here. The red being continued exposure to these sounds can cause hearing loss, so you better get some hearing protection. So what's happening here, as the sound wave energy is transmitted to the stapes, it's just tilting. And because the inner ear structure is fluid filled, 100% all the way full, that sound wave energy is then transferred. It vibrates the fluid, which can then deflect structures that have the organ of hearing, which we'll get into later. But this also shows this oval window versus this window here. That's called the round window. So don't confuse oval window with round window. Round window is just, it's absorbing that energy that the oval window is transmitting. Absorbs energy being transmitted to oval window. So if this deflects in by the rocking of the stapes, the oval windows rocking in, there's fluid displacement, which will make that deflect out. So this will kind of like be rocking back and forth, and this will oscillate with it, okay? Because fluid energy, it's continuous, and it needs somewhere to kind of like end up. All right, questions for you. Be right back. You work on those. Right, number one, the components for outer, middle, and inner ear. For outer, it's the pinna and the auditory canal leading up to the tympanic membrane. For the middle ear, it's the auditory ossicles, malleus incus stapes. They're taking those vibrations from the tympanic membrane. And also I noted the auditory tube, not to be confused with auditory canal. And then we have the inner ear structure itself that receives input from cranial nerve 8. Let's look at the inner ear structure next. It's called the vestibular apparatus. It also has the cochlea. But here's how it sits in the skull. It's inside the temporal bone in the temporal region. I already told you that. If we kind of saw the skull cap off and then look down on the cranial base, that's this view. So here's the cranial base. That's foramen magnum, where the brainstem exits. There's the petrous region of the temporal bone. This is the part of the temporal bones that kind of stick in. Okay, kind of like that. You can see the inner ear structure inside there. There's cranial nerve 8, and it exits the skull to get to the inner ear structure via the internal acoustic meatus. So write that down. So cranial nerve 8 exits internal acoustic meatus. Remember, we're in the temporal bone, petrous region. To innervate, cochlea, vestibular apparatus. So that cochlea, vestibular apparatus, are simply what we've been calling the inner ear structure. If you were to do a dissection of that and just take that and just kind of scoop out this inner part there, you would see, talking about the cochlea, this kind of arrangement, all these like little triplets of chambers inside the cochlea, which is what I want to talk about next, which is the special sense for hearing, really. Equilibrium is for the vestibular apparatus. So the cochlea is this part. So I like this picture. We can see both branches of the vestibular branch and the cochlear branch, and how the whole inner ear structure is organized. So to simplify that, let's draw it here. So the cochlea is this kind of snail-looking thing, and it has this big vestibular part with these three semicircular canals. So basically cochlea, the vestibule, and then you have 1, 2, 3 semicircular canals. So those are the basic parts of what we call the inner ear structure. Cochlea, vestibule, semicircular canal. These all convey different sensations of hearing and equilibrium. But also I wanted to define what's meant by bony labyrinth and membranous labyrinth. As you can see, like if you could see on your slide, it's got this inner membranous labyrinth and it's surrounded by this outer bony labyrinth. So because this is such a complex shape, let's take a different shape so we can understand how the membranous labyrinth is inside the bony labyrinth. Membranous labyrinth inside a bony labyrinth. Labyrinth is a complex network and it's like a maze, which this is. But if we took this, let's say we just cut at any point. If we just cut and look at the cut circle, we would see inside a membranous labyrinth in blue, and it's filled with a fluid called the endolymph. Pretty much a variety of blood plasma. And surrounding it is the bony labyrinth, it's the bony part, and it's filled with a perilymph. So endolymph, perilymph, these are the fluids that fill the inner ear structure. Now let's look at the cochlea a little more. If we were just to remove the cochlea, here's what we see. Let's remember our orientation here. Footplate of stapes, oval window, round window. But let's take the cochlea, let's unwind it and chop it right there so we can see one of those little triplets. Just to remind you, that is basically one of these chambers there. And here's one of these chambers just drawn big. And you can see how the nerve fibers are kind of going in to innervate this organ of hearing right there. So this organization is divided into 3 chambers or scala or ducts called scala vestibuli, scala media, or scala tympani, or you could call it vestibular duct, cochlear duct, tympanic duct. Okay. So basically what you got there is that 3 chambers: scala vestibuli, scala media, scala tympani. So what you see here is the organ of hearing, and what you see are these hair cells surrounded by 2 membranes, a basilar membrane and a tectorial membrane. So I have some more pictures here. I do want you to know that the scala vestibuli and scala tympani are both filled with the perilymph. And that the scala media, the cochlear duct, is filled with an endolymph. And that the organ of hearing are actually these very important hair cells. Right there. They're drawn in blue there. Let me get a blue pen. And they're sandwiched between two membranes. This bottom membrane is called a basilar membrane. It's a more flexible membrane. It's bendy. You can deflect it. It varies. Sometimes it's less flexible, sometimes it's more flexible. We'll get into why that's important. But above the hair cells is a very stiff tectorial membrane. And let me advance the slide so you can see it labeled. There's outer hair cells. Let me draw an inner hair cell there. Well, anyways, they're all covered by this stiff tectorial membrane going over the top. Tectorial membrane, it's more stiff, not as flexible as the basilar membrane. And so all of these hair cells, the inner and the outer, they're connected to the cochlear branch of cranial nerve 8 for hearing. And these cells, when they're triggered, they're mechanotransducers. In other words, something physical has to make them fire. And it's a physical moving of that hair cell bundle. If you just wiggle those little bundle of hair cells, it'll make this cell fire and communicate with that nerve ending there. And that's how this works. This is the organ of hearing. The basilar membrane, the tectorial membrane with the hair cells in the middle is considered the organ of hearing, and we call it the organ of Corti. Okay? And those inner hair cells, they're actually really important. Even though they're outnumbered by the outer hair cells, they're transducing about 95% of the sound that you hear. So 95% is for those guys, the inner ones. Okay, that's a 5, 95%. Let me write that again. So the inner versus the outer, okay? And let me write the term on the board. Organ of Corti. That's what we call this thing. The organ of hearing, organ of Corti, that's for hearing sound. That's the sensation, sound for cochlea. So the hair cell is important. Because that hair cell is found all throughout the inner ear structure, and that is the cell that is responsible for hearing and equilibrium. So more on hair cell later, but for now, just appreciate that the hair cell looks like that, where it's a regular cell, that has these special cilia at the top. Turns out they're all connected, these little tips. These little tip links are all linked. And if you just kind of move the hair cell bundle, that is what triggers this cell to fire on a nerve ending of cranial nerve 8. So what's remarkable about this cell is it's a cell of communication. And we talked about that idea before in neurons, where neurons are cells of communication. They have areas of input and areas of output, the dendrites and the axon. Well, the hair cell is a cell of communication, but it has neither. It has no dendrites, no axon. Hair cells are cells of cells of communication, but have no dendrites, no axons. But it still has an area of input and output. So here's the area of input. Just wiggle that apical hair cell bundle. That's your area of input. Now to communicate down here. So down here is your area of output. So it's still able to function as a communication cell because it still has area of input and output. So again, these are an organ of Cordia and everything else, as we'll see. Here's a picture of just those little hair bundles. They give you numbers of inner hair. You have way more outer hair cells. The inner hair cells transduce 95% of sound, whereas active movements of the outer hair cells, they help to amplify the signal. I thought that was a nice picture of inner versus outer hair cells. The next topic is pitch discrimination. So this is still our cochlea, but it's unwound into a straight tube just so we can kind of see how it's organized here. And the idea of pitch discrimination starts to deal with how we distinguish high frequencies like treble versus mid versus low frequencies like bass. So think of sound as sound waves, and those sound waves have energy. Like just a typical wave of sound. Now sound waves travel, okay, and they travel in waves, and basically sound wave energy, think of that in terms of the length of the sound wave and the magnitude of the sound wave. Start with magnitude. If the sound wave is large, we perceive that as loud. Magnitude, or sometimes they call it amplitude, just from this to there. That would be loud. But this would be soft or quiet, okay? Just big versus small. That energy is perceived as loud versus soft. But then there's the wavelength, okay? So magnitude and then wavelength. Wavelength is like if you have large waves, okay, so say from like there to there versus short waves from there to there. These longer waves have less energy and we perceive them as lower frequencies, okay, like bass. So this one here, less energy, low frequencies like bass, okay? But this would be from there to there, more energy, higher frequencies like treble. So that is what we call pitch discrimination. High-pitched sounds, low-pitched sounds, depending on the wavelength. Less energy, more energy. Now we can start to unpack this picture here, how the cochlea is organized. And one thing, one more definition before I draw that on the board here. We keep saying frequencies. We need to find that. Frequency is usually, well, it's how often a wave passes a fixed point. In this case, we're talking about sound waves. So how often a sound wave passes a fixed point. Okay, so let's say that's the point. And if you have low sound wave energy traveling in that direction, you have less frequency because they're longer. It's going to take longer for a wave to pass a fixed point. And so we call it frequency. We measure in hertz. I usually just abbreviate hertz in Hz. That's how many waves per second pass. So if it's a lower frequency, say 100 to 20 hertz, we call those low frequencies. We perceive that as bass. But if it's a higher frequency, more sound waves will pass per unit time. And then so higher frequencies could be like 3,000, 4,000 hertz. And we perceive that as a higher pitch, like treble. Okay. So in understanding how that plays in here, you know what the hertz mean now. Higher energies, lower energies. Higher energies for higher hertz, low bass frequencies, less energy. So you have to associate the hertz with the energy. High energies can hurt your ears, but lower energies maybe not so much. So let's kind of draw this out. Cochlea. So on this end, here's my stapes. Okay, there's stapes pushing on the oval window. And let's say it pushes it in, oval window being pushed in. So that means you must push out the round window. You push in, you push out. And what's happening is you're transmitting this sound wave energy to this chamber up here. This is the scala vestibuli, or the vestibular duct. the vestibular duct is actually, when you get to the end here and go back, this is actually the tympanic duct. And this middle row here, that's your cochlear duct or your scala media. Cochlear duct right there. But it's in the cochlear duct that you have all of your hair cells. Let's say you got a hair cell here. And you can pretend you have basilar and tectorial membrane in it. Hair cell here. And put a hair cell down here. These are my hair cells in the organ of Corti. And what they call it here, when you get close to this end, they call this the base, B-A-S-E, of the organ here. And when you get closer to this end, closer to stapes, the basilar membrane is more stiff. So let me draw in basilar membrane. I can't remember what color I used before. So my red line is my basilar membrane. I'll draw it right there. The thing you got to know is when you get closer to this end, the fibers of the basilar membrane are longer. So let me just draw longer membranes. It's usually illustrated like this. But as you proceed out here, the fibers get progressively shorter. So it's kind of like it takes on this pattern there. So this membrane, I drew it as a straight line. Really, the fibers are getting shorter and shorter and shorter. And that's really important because as the fiber gets shorter, basilar membrane becomes easier to deflect. It's more flexible here on this end because the fibers are shorter, less flexible, more stiff on this end, less flexible. That means it takes less sound wave energy to deflect here, takes more sound wave energy as you proceed this way toward the base here. Okay. And so let's say you're hearing bass, 1,000 hertz. 1,000 hertz, when it's introduced into the vestibular duct, it's going to move along the cochlea. And because the fibers are less flexible here, it's not enough energy to deflect it. So it'll just keep going down and down and down and down until it gets to the first place where it can deflect it because it's more flexible here. It'll push down and it'll deflect down on the basilar membrane. Okay, and it'll oscillate at that frequency. So imagine these windows here, one's pushing in, one's pushing out, okay, at that frequency. It'll eventually die out. Okay, it's like twining a rubber band. The rubber band's easier to twine here, so low energies travel further because it can't vibrate them when it's more stiff. Okay, so it has to go all the way down to here. Maybe that's 1,000 hertz. Maybe that's bass. And I have a link to a free hearing test, so you can kind of see what I mean by that. Let me go to that. So let me just go answer the questions here. For computer speakers, cool. Okay, that's cool. Let me adjust my... Alright, I can hear that tone. Hopefully you can hear it at home too. And this is a hearing test. I ask you all these questions. You can do it yourself. And well, my hearing's okay, so I'm going to answer no to all of these. All right, so 1,000 hertz tone. So when you hear that tone is 1,000 hertz. So it's traveling down and it's stimulating these hair cells, which transmit that tone. So these cells being stimulated is what you just heard. That's the sensation, pitch discrimination. And let's move on. I heard that. Here's 2,000. Well, that's getting higher pitch, so more energy. So it's for the 2,000, it's going to travel along, and maybe it's going to deflect sooner because it's a higher energy, so it can vibrate hair cells in an area of the basilar membrane where it's less flexible. Okay. So I heard that. Let's proceed. Here's 3,000. Cool, I heard that. Here's 4,000. Next question. Well, I passed the test. Okay, great. Let's go back to the PowerPoint show. So what's happening here, as you heard the higher and higher peaks, the lowest one that I started with, low sound wave energy, it can't vibrate here towards the base, B-A-S-E. But as you get further out here, then it can vibrate. Because out here, ooh. Oh man, I'm sorry. I drew this backwards. I should draw it like, this made me realize I drew it wrong. So mistake, oh, sorry. Had a brain fart. It's actually like this. Like this is correct. I drew it wrong. Sorry. Erase your notes. That was right, but I just drew the fibers wrong. Longer fibers, more flexible, easier to deflect. Shorter fibers, stiffer, less flexible. So short fibers, less flexible. Longer fibers, long floppy fibers, more flexible. Okay, so this end more flexible, this end less flexible. The fibers are shorter, they're stiffer. As they get longer, it's easier to deflect. Shown correctly here. Anyways, longer fibers, easier to deflect. When you do deflect it, the low frequency sounds can deflect it. It can't deflect anywhere before, not enough sound wave energy. But as the sound wave energy increases with higher hertz, you can deflect sooner. That's mid frequencies, high frequencies, it deflects first. And so these are never stimulated. Now what happens if it's so low, less than 20, that it never deflects anything? It just goes completely all the way around and you hear nothing. Okay. Questions for you. The oval window transmits sound wave energy to which scala first? Right here, scala vestibuli. Which scala has the organ of hearing? Right in here, scala media or the cochlear duct. What do we call this organ of hearing? The organ of Corti. Describe the structure of the organ of hearing. The organ of Corti, know the two membranes. Know the tectorial membrane, more stiff. And then you got hair cells with the hair stuck in it. I'll draw a couple there. I think of this as a hair cell sandwich. And then you have the basilar membrane at the base, underneath it. And this is progressively less flexible to more flexible. Again, that's my mistake there. So that's this picture here where it's less flexible with shorter fibers. As it gets longer fibers, more flexible, less flexible. Short, long fibers. That's basilar membrane. That's the thing that's deflecting, because the basilar membrane is flexible to an extent, but the tectorial membrane is very stiff. And that's important to have flexible, stiff. So the bottom membrane is vibrating, kind of like vibrating. That'll stimulate the hair cells to wiggle back and forth. So that is what is causing the physical movement of the hair cells. The flexible membrane vibrates, the stiff membrane stays still, and that is what is able to wiggle that hair cell bundle at the top. So that answers number 3. The picture to the right, which depicts the low frequency or bass sounds, top, middle, or bottom? Bottom. Moving on to vestibular apparatus. Shown here. So you got utricle and saccule inside there. So we're done talking about the cochlea. And then so in this vestibular part, again, here's cochlea, all wound up like a snail shell. And the vestibular apparatus, now I drew them separately, but they are connected. So inside there, you have this kind of subcompartment inside the vestibule. Vestibule is a space, and inside there you kind of looks like this. Utricle. Saccule. This is the organ for equilibrium. And inside utricle and saccule are these little structures called the macula. M is for the macula. And it's the macula that has the hair cells. Here's a picture of the macula, shown on the slide there. So this little structure is this structure there. And so what we see for the macula is that the hair cells are now embedded in this otolithic membrane. What we'll see is that these hair cells of the macula are for detection of like linear acceleration. Like when you accelerate on the freeway, go from 0 to 60 in your car, you feel that sensation. or the big drop in a roller coaster, and you feel that linear acceleration of the roller coaster, making it very exciting. And so macula, draw a hair cell here. Here are the little hair cells, and the tips of the hair cells are all linked, and they're stuck inside an otolithic membrane. So I'll use red, or how about orange? Try another hair cell over here. Otolith membrane. That means tiny little rocks. So there's actually little rocks called otoliths on top of the otolithic membrane. Let me draw some like little rocks on top of here. Like little grains of sand that kind of move back and forth. And as they move back and forth, the otolithic membrane will move with it and stimulate the hair cells, which are hooked up to the vestibular branch of cranial nerve 8. Otoliths. So otoliths are the key to the macula, because when you live on Earth under gravity, those rocks, they settle down on top of the otolithic membrane. As you move your head back and forth or accelerate in your car and feel that, those otoliths slide back and forth, stimulating the hair cells, which are hooked up to the vestibular branch, cranial nerve 8. Vestibular branch, VIII, that's cranial nerve 8. So that's how you stimulate the hair cells there. So again, a little bit more about those hair cells. They have channels directly gated by mechanical stimuli. That mechanical stimuli is the wiggling of the hair cell bundle. When you stimulate the hairs, quote unquote, of this cell, really they're kinocilium and stereocilia. You have more stereocilia, but that kinocilium, you have one, large and tall, and I drew it as like a little dandelion, the first one here, the kinocilium. Okay, that first one there. The rest are called stereocilia, many more. When you stimulate the hairs, you depolarize the cell. And so here's the little hair cell bundle. There's the cell, there's the hair bundle there. That's what we saw before. Here's the illustration. This is really nice. See that little kinocilium right there, the little dandelion thing? And then you can see the stereocilia. They make a little apical hair cell bundle at the top. In experiments done in the late 70s by Hudspeth Corey, they used the macula from frog. And so know these guys. Hudspeth Corey, 1977. They studied the hair cell from the saccule of frog. I believe it was bullfrog. That was their model. Use saccule. Bullfrog, I believe. Anyways, they removed the otolithic membrane, allowed them to get their little glass electrodes, allowed them to stimulate. And when they wiggled back and forth, they detected depolarization, hyperpolarization, right there. That's what I say there. When the stimuli moved in the direction toward the kinocilium, it depolarizes, and move it in the opposite direction, it hyperpolarizes, and that's the stimulation we're talking about. What they hypothesized, the reason why you had excitation versus inhibition, they called it the tip links hypotheses, that all the tips of the hairs were actually linked. So that's what I want you to know about Hudspeth Corey. He came up with what's called the tip links hypothesis. So know that hypothesis shown here. When the tip links stretched because you move it towards the kinocilium, that causes the depolarization. So you depolarize those hair cells when you move it toward the kinocilium. When you tilt it the other way, the tip links are slackened, producing an outward current that we call inhibition. Depolarize, hyperpolarize. Here's another picture showing you the hair cells. Otolithic membrane, tilt head one way, depolarize, tilt head the other. They slacken, and the depolarization, they show you the action potential frequency there, just moving your head. Okay. So we say that you're detecting a linear acceleration for the macula. So let me write that on the board. So the vestibule, with their utricle saccule, with their macula, that detects a linear acceleration, like tilting your head or going from 0 to 60 in your car. That's the quote-unquote equilibrium sensation detected by this inner ear structure. And let's move on from the vestibular apparatus to the semicircular canals right there. You have three. Just call each of them semicircular canal. They're oriented, each one, in one orientation for three-dimensional space, like horizontal, vertical, and an oblique angle. So know your three semicircular canals. If I were to just draw one, I'd draw it like this. The base is called the ampulla. In the figure there, I call it A. And the other thing, SC, from there to there, is the semicircular canal, right there. So SC is semicircular canal, A is the ampulla. Now the ampulla has the sense organ in it, and it's called the crista ampullaris. So let's look at it. Let's look at this on the next slide. So this is that. So what we see there is you have this structure here. There's a cupula. I think of a 70s lava lamp. It's a gelatinous structure, and that's got the hair cell stuck in it. Okay? So got a little cupula. Draw a little hair cell in there. And it's stuck in a little cupula. But the membrane that the hair cell is in, call it crista ampullaris. It's the crista ampullaris that has the hair cells. And this organ will detect rotational acceleration because there's fluid in it. And the only way to get the fluid to move crista ampullaris, is to rotate. So these three semicircular canals will sense rotational acceleration. So they should give the example of a figure skater spinning around. The fluids move one way. Again, tip-lick strange. Then you stop, but the fluid still moves, and you still have the sensation that you're spinning around. Because you're moving it the other way, you sense that you've stopped, but you still feel like the room is spinning. Okay, because you're still moving the hair cells one way or the other. So depolarization, hyperpolarization, and all right, questions for you. We'll end with this. Name the structure for each of the following that contains the hair cells. It's a good review. For the cochlea, organ of Corti. For the utricle, saccule, macula. Semicircular canals, it's that crista ampullaris with the cupula. I'm going to stick with crista ampullaris. Those actually have the hair cells. In terms of the sensation that's detected, for the cochlea, it was hearing. We did pitch discrimination and all that. For the macula, it was linear acceleration. For the crista ampullaris, rotational acceleration. All right, that concludes the ear lecture. Thanks for watching.